So we're going to talk a little bit about amino acids and how they actually come together to make proteins. So up here right now I have 4 amino acids, and the first thing you need to know is actually their properties, right? So these 4 amino acids, they have different properties based on their R chain, which is shown from here, right? So all of these have different R chains, R groups. Now, the way you tell non-polar R groups from each other is that they all have carbons and hydrogens. And because of that, the electrons are shared evenly between them, and therefore there's no polarity in the R group, right? So now, both of these have carbons and hydrogens in their R group, and therefore there really is no polarity that can arise, and therefore are non-polar. Whereas this molecule right here has this OH group right here. Now, because oxygen is more electronegative than hydrogen or carbon, it has this pull of electrons, right, on either side, and therefore making it have a partial negative charge, and therefore making it polar, allowing it to have polar interactions. Now, if something has, if an R group has nitrogens in it, nitrogen has the capability of attracting hydrogen atoms out of solution, right? So nitrogen has these lone pairs right here and can attract the hydrogen and then essentially form this. And I think your book actually shows arginine in this form. But anyway, because nitrogen can take hydrogens out of solution, it's going to be basic. So anything with the R group that has nitrogen will be basic. And on the other hand, if you have an R group with a carboxylic acid group, right, which looks something like this, this carboxylic acid group can actually donate this hydrogen right here into solution, therefore making it acidic. Right? So if it donates this hydrogen, it's going to end up looking something like this, which is actually how your book shows it. So anything that has this group is therefore acidic, anything with nitrogens is basic, and so on. Alright, now let's talk about actually how these proteins get formed from amino acids. Right? So we have 4 amino acids up here, right? And we'll write them out. Let's see, so this is phenylalanine, and I believe Professor Ashmore wants you to know the 1-letter abbreviations. So phenylalanine is abbreviated with F, and this is alanine, it's abbreviated with A. This is arginine, abbreviated with R, and tyrosine, abbreviated with F. So if these 4 come together, they'll form our favorite protein called fart. Now, how do they come together? So now, this is formed by dehydration reactions. Right? Now, as you can see over here, if you release a water molecule from here, right, you have a full bond going all the way through. This is known as a peptide bond. You get a peptide bond here, another dehydration synthesis right here, release of water molecules. The same thing over here.
So what Richard's drawn out for you here is the primary structure of this FART protein. This FART protein will continue to fold in on itself and create a secondary structure, tertiary structure, and maybe even a quaternary structure. So for this FART molecule, this FART protein to fold in on itself and create a secondary structure, what happens is you have these H's and these O's in the backbone. And notice I'm just talking about the H's and the O's in the backbone for now. The H from here, let's say, this backbone H, will make a hydrogen bond with this H, with this O, let's say. And this O can make a hydrogen bond with this H, right? So you can have all variations of hydrogen bonds between O's and H's in this chain. You can have one over here too. Or we can just leave that one hanging. It doesn't matter, it just depends. That's what makes the proteins vary in their structures and in their functions. You can have all kinds of different interactions taking place to create different types of secondary structures. Secondary structures, once you have these hydrogen bonds in place, secondary structures can either become alpha helices, or you can have the beta plated sheets, right? You can have the sheets. And really, whichever one is created just depends on what kind of interactions happened up here in the backbone. It could be anyone. Tertiary structures arise from the interactions between these R groups now. When we go from secondary alpha helices and beta pleated sheets to tertiary, we have interactions between these R groups and these R groups and all the other R groups here. And so what happens is you could have hydrogen bonds again if you wanted. Like maybe this protein will decide to have the H's from here. Maybe this H will hydrogen bond with this O over here from this tyrosine. But the point is that the hydrogen bond that happens is between R groups. It's not the backbone anymore for tertiary structure. On top of that, you could have interactions like you can have ionic bonding between this alpha helix and this beta pleated sheet somewhere. You could have hydrophobic interactions, groups that are non-polar interacting with one another and staying away from the polar areas of the surroundings. Or you can maybe even have disulfide bridges be created. The FAR molecule, because of the R groups that we've chosen, does not have sulfur in it. But let's say it did for a second. Let's just say it had a sulfur here, and this sheet has a sulfur here. Right, so these sulfides can come together in a disulfide bridge, and this is the strongest of all the interactions that can happen when creating a tertiary structure. So what we have here now is this whole thing is now connected. The beta pleated sheet is connected to the alpha helix. You can have another alpha helix here. You can have a sheet going right through here. It all just depends on the functions and properties of this protein that's going to be created in the end. So quaternary structure will arise from interaction between this big subunit here. We have all these alpha helices, all these beta pleated sheets come together. This creates one subunit of this FART molecule. But if we want to create a bigger FART protein, this subunit, there's going to be an exact copy of this subunit down here. Let's just draw it like this for now. Let's say this is the exact copy of this, right? These two can interact the same way that tertiary structures are created. You know, you can have like a disulfide bridge, like we were saying before, maybe, between these two to connect them, and now it's one big protein. We can have, you know, hydrogen bonding between these. You can have H here, and you can have an O here. You have hydrogen bonding between those. Anything that happened within the tertiary structure to create the tertiary, those same interactions apply for quaternary structures and creating them. It's just a bigger protein now.